Again, thank you everybody, and good morning here, and I guess good afternoon. You can sorry, again about sort of the schedule. So today is actually the first day of our semester starting, so it's a little bit different. So my topic of the presentation is 3D bioprinted vascularized tissue model for cardiovascular and cerebrovascular applications. And I'll be focusing more on the cardiovascular applications, and I'll just throw a little bit of slides at the end. Because the cerebrovascular work is more on our initial stages. So a little bit of background about 3D bioprinting. I know there are one or two topics already presented, but still I'll give you a little bit of overview. It's a technology to develop, cell-level complex tissue constructs. And one of the important components is that you need to have bioink materials to form the extracellular matrices. And then the right composition of bioink is very critical, because once you encapsulate the cells in it, the cells has to proliferate and have the right native environment for them to grow. And then also when you bioprint, you put the cells to a stress. So you have to make sure you have to optimize a lot of the bioink components as well as the printing parameters to get a nice native tissue. So that's one of the main challenge of 3D bioprinting. And once you print tissues, that's great, but how you can make sure that the tissue will be viable for an extended period of time? So one of the main challenge in tissue engineering bioprinting is vascularization of the tissue, because you have to make sure that you can maintain the flow of nutrients and everything to the tissue. So you have to make perfusion channels and network in order to maintain the integrity of the bioprinted tissue. So there are a lot of challenges in a bioprinting. It's still a sort of a newer technology, but a lot of advances has been made in the past few years to bioprint tissue constructs. So this sort of gives you a little bit of a overall what I also mentioned, what are the important elements involved in a 3D bioprinting. As you could see, the extracellular matrix, the cells, the vasculature, and then the different types of bioink which you use, because all this will play a role into cell-to-cell communication, proliferation, differentiation, migration, apoptosis and metastasis. All of these are needed for a real 3D tissue native environment, which you need to sort of develop. So as I touch base, that there are a lot of parameters which will actually affect your bioprinting process. And when you bioprint, it's basically we are using, say, extrusion-based printing mostly. So you have a pressure being created. So you have to control those, extrusion pressure. Then you can use different diameters of nozzle through which your extrusion material or the bioink is coming out. So that is going to affect your shear stress. So that's another important thing. And then the cells will be already mentioned, will be experiencing shear stress during the bioprinting process, which will also impact your cell viability. So you need to really also look at the rheological properties, because the bioink is the one which is going to produce the native environment. So you need to look at the interplay between shear stress, viscosity, and other rheological materials to see how these are impacting the ECM matrix and thus the viability of the cells during the printing process. So a lot of parameters come into picture. So why we focus on some of these applications? As I said, I'm focusing on the cardiovascular applications, because in a normal endothelial function, your vascular function balance is mechanism is balanced. So that's why you have a mechanism leading to contraction and relaxation. So when there is a dysfunction, then you could see there'll be a disbalance between vasodilation and vasoconstriction. That's the problem which happens during the cardiovascular disease. And I'll not go into the very detailed of the cardiovascular disease. As I said, I've been talking more of the bioprinting, just a little bit of a perspective. What are the things we're looking at? One of the components of a cardioprotective roles is nitric oxide, because we'll be looking into some of these things. So I will give you a little bit of background. So nitric oxide is a pivotal signal messenger in a cardiovascular system and plays an important role to against protection against the onset and production of cardiovascular disease. And in a healthy condition, your NO will be a major endothelium-dependent vasodilator, which is found in our body. And this is sort of a maybe complex diagram, but just from perspective, I'm not a biologist, but we also have to learn the basics of it, because to understand the mechanisms. So we could see that there is a production of angiotensin 1 will lead to production of angiotensin 2, and that will tend to sort of increase your blood pressure and it will turn off the initial stimulus of angiotensin 1, which sort of a protective role. So you can use some blocker, and we'll talk about a little bit. We started working on the conclusion. I'll talk about it, that how can you use some blocker like losartan to prevent the formation of angiotensin 2, which sort of is not good for cardiovascular disease. So we are looking at two primarily assays, nitric oxide and reactive oxygen species, because an imbalanced production between these free radicals will result in oxidative stress. So we know in a vascular disease, you're going to have reactive oxygen species generation, and that's going to be increasing. And then your NO availability is going to be decreasing. So we'll be looking at, that's the reason we chose these two particular mechanisms to look at how the oxidative stress is sort of affecting the cardiovascular disease. There can be many other pathways. As I said, we are looking at some applications, which I'll talk a little bit later too, but these are the two mechanisms we are looking at it. So as I mentioned that, again, that in vascular disease, ROS production will increase and your NO availability is going to decrease. And this is sort of how you could see that angiotensin 2 can sort of increase, can leak into problems with the cardiac disease. So you have to maintain the balance between NO and ROS productions. So that's sort of a background for motivation for looking at cardiovascular disease. So what our goal is to actually develop extrusion-based bioprinting to 3D print a vascular tissue and perform a parametric study of the ECM matrix so that you can enhance the tissue functionality. And one of the main things is that also, when you do bioprint, you actually have to maintain them for an extended period of time. I'm presenting results for 15 to 20 days, but right now you're actually making it for 30, 45 days so that you can maintain the viability of them. And then, of course, you have to do some characterizations of the cells once you bioprint them and see the viability. So that's sort of our overall goal of this work. So this is the bioprinter we are using. It's a CellLinks 6-head modular bioprinting. That's a lot of advantages, because by using these multiple heads, we can sort of involve multiple cells at the same time. We are right now just using only 2, because we are using 2 types of cells, one for the matrix and one for the endothelial and for the vascular channels, endothelial cells. But it, as you could see, it has got a temperature control. It's a lot of optimization is needed. I mean, it's not really just pressing a button, you're going to get a print. So that's one of the really challenge, that you have to optimize you could see the temperature of the print bed, then the nozzle shape, and whatever nozzle size you're using. And because a lot of times what happens, like once you prepare your bio ink, by the time you start printing, these things can harden right away and you not get enough extrusion. Then you have to put a lot of more pressure, and that's not good for the cell. So there's a lot of optimization you really need to do when you're doing your bioprinting. So again, cell mixing, bioprinting, and cross-linking is another important thing that's needed. So just like a quick overview. So basically use a solid, what is a bioprinting technique? If you're not familiar, you create a, STL file, and there are different programs you can use. We use, for example, Creo. And then you can use these G-code manipulations if you want to do a lot of manipulations of what you really want to do with your design. So you have to have a little bit of familiar with the G-code, and then you export that to the bioprinter. But these days you can actually get a lot of custom, like an open source G-code that you can use for bioprinting. So again, as I mentioned, the rheology is an important part. So we did some numerical actually modeling so that you can optimize the nozzle size, pressure, and the printing parameters, because the viscosity, surface tension, and all of these things are going to play an important role in calculating the shear stress, what is involved, which is going to affect the bioprint quality. So this sort of tells you some of the parameters which you have to optimize during the printing. I'll not read it out, but like the needle diameter, the print speed, the printed temperature, the print bed temperature, the layer height, then cross-linking frequency, because you have to cross-link it, because once you print with the bio ink, unless you cross-link, it's not going to form a right matrix. So you can sort of cross-link. Timing is again another parameter. So all of these parameters, what you could see, have to be optimized to get the right ideal print for your application. So a lot of optimization is needed. So this sort of gives you a little bit of a G-code line format, what you have to manipulate. Just taking a perspective, what are the parameters you can manipulate? Your locations, and then your translation rate, your extrusion rate, your pressure, then what type of print head you are going to select. So these are the sort of the layout of a G code what you have to have before you set the printing. And as I mentioned to you, the printing of the vascular channels is the most important component. So we are using a sacrificial way. That means you first create these sacrificial channels, and then you first print this sacrificial channels, as you could see, and then you put your biomatrix, extracellular matrix, and then this material is made of pruronics. You can flush the channels out. So in a sense, then you'll be left with a channel inside the tissue matrix. So that's the technique is known as sacrificial matrix. As is mentioned, is you remove them so that you can leave aside a matrix with hollow channels, basically. And then you line the channels with endothelial cells to form your vascularization. So you're going to have a cells and a matrix, whatever you want to put, fibroblasts, cardiomyocytes in your ECM with the bioink, and then your channels will be hollow, which will be putting endothelial cells to form your vascular network. So that's sort of the overall goal of a 3D bioprinting. And as I said, sacrificial bioprinting, there are many other ways you can do it, but that's one way we feel is very important because it can help you to sort of create the right geometry, and you can endothelize this the tissue matrix so that you can provide the nutrients and to these channels. So here are some of the bioprinting constructs, what you could do, what we printed, you could see here. This is sort of our matrix, and these are the channels, and we do some, and we are using some photogel. Commercially right now, Gelma, one of the things we really like to do is to make our own bio ink, but initially for optimization, it's better to buy, it's a little more expensive, but write the bio ink from the manufacturer of the bioprinter. So using photogel, which is nothing but the methacrylated gelatin materials, which you're cross-linking, and then you have to optimize all of this, as I mentioned to you, a lot of these parameters like cross-linking time, the photoinitiated intensity, all of these things, and we are using pruronics, the sacrificial ink. So you could see how they are measuring this, creating these channels, and you are then filling them with endothelial cells. So this is your vessel channel, and this is your matrix. So this is sort of our basic approach. And we are looking into many applications which I'll do later on. But for example, we are going to make it in a perfusible bioreactor for some space applications. So this is some of the bioreactors which we have provided, and we're using again Gelma, as you could see in this. So you're able to print in this sort of a mini bioreactors so that we can use it for applications. So now I'll go over quickly about some characterizations, what you have done. We did rheological studies, we did viability, then biocompatibility, some qPCR, and then our nitric oxide. As I told you, we're looking at some applications for the reactive oxygen species ROS, and then morphological analysis. So rheology is one of the important part of a bioprinting technique because it will help you to understand the viscosity of the gel and the shear thinning properties, which is very important component. So you could see how the shear rate and the viscosity sort of varies for the bioink we are using. And here we are looking at different Gelma concentration to sort of optimize our pinking, because what you could see, the viscosity is going to affect the amount of gel which you can extrude during the overall printing process. And you're using this particular rheometer to do the rheological analysis. And then the other thing also have to measure some of the rheological parameters, the shear storage modulus, which is also another important parameter. And as you could see that how the cross-linking time is another important parameter, because your shear modulus is going to be a function of the cross-linking time. But you could see that after a certain time, it is not a major parameters. So depending upon what type of viscosity or bioink materials you want to sort of need, you have to play with your cross-linking time to get the right shear storage modulus. So then for the viability of the cells, we are using a classical live dead assay from in vitrogen kit. It's basically that it stains the cells with calcine stains of the green, which is the viable, and then the ethidium homodimer stains, which are red, are your dead cells. So you could see that we hardly got any dead cells. Maybe there are one or two you could see here. And one other important thing is also your perfusion parameters. So we want to optimize that. So which is this is sort of a best optimized, we call it, in terms of the most viability. It may be not hard to see. You could see here there are some more red cells. So the perfusion inlet flow velocity of your perfusion parameters, another how you're putting the nutrients at what velocity. For example, we did a lot of mathematical model, as I mentioned to you, to calculate the viability and the shear stress involved. So this is just one example how your inlet velocity to the channels can impact your cell viability. And then you also look at MTS, which is a calorimetric method for detecting also the number of cells by looking at the production of formazan, and the formazan is directly proportional to the number of viable cells. And you look at the absorption change at 490 nanometer using a spectrophotometer. And so here is some, then we also did some RNA extraction because we want to look into some analysis how these bioprinted tissue constructs behave in an environment with different types of cells. So we extracted the RNA, and then you perform qPCR, and we basically put this bioprinted with some young HUVECs and some old HUVECs to look into the characteristics of the environment, how the bioprinted matrix cells can survive in an environment when it is exposed to older HUVECs and newer HUVECs. And we are looking at some microRNA. And we used in a qPCR, you have to select always a housekeeping gene. So in this case, you used a microRNA 16, and we're looking at some mu146 and mu217 are the two microRNAs which we have expertise in and looking into it. So this is something, as I mentioned, that we are looking at the ratio with the old and the young HUVECs. You could see some of the numbers, and I'll show you. So you want to make sure that you have good quality RNA so that you can do the PCR. So here is the data of a qPCR. We are looking at the two microRNA, how the expressions happens when you are exposing this bioprinted. So what we did, we took the bioprinted matrix in a perfusion in a chamber, and then we also let the constructs be surrounded by old HUVECs and by new HUVECs in another environment, in two different controlled environment. And you wanted to see how these HUVECs, old versus new, when they're co-cultured, are sort of show the manifestation of the different microRNA expressions. And you could see here for microRNA 146 how there's an increase when old HUVECs, and here there's a decrease. So you could see how the different types of old and healthy newer cells of HUVECs were basically influencing the microRNA expression from these bioprinted constructs. So these are all very important tests. You want to make sure that these constructs represents the true native environment. And then we are looking also at nitric oxide assay. As I said, that's an important assay for us to look at a lot of our applications, in terms of oxidative stress. I'll not go into the details. So, but one thing I want to just mention what we really did. When you do this sort of bioprint, you want to make sure that what you are really going to see, the mechanisms are not an artifact. So what we did, we used some agent to increase the NO production and want to suppress it. So you could see with LN is used to inhibit the NO, and then sodium nitroprusside is used to increase. So when we put a bioprinted construct, we want to make sure we get the true manifestation of the mechanism so that these tissues are behaving like a real tissue environment. So this is sort of a basic nitric oxide assay. What you do, you basically fix it in a vector shield, and then you look at the microscopy. So this is the control. And as I mentioned to you, that if you use sodium nitroprusside, which is an enhancer, you're going to see that you're going to get much more brighter because your NO production is increased. And then LNM is an inhibitor, so it brings it down. So all these characteristics we did just to make sure that our bioprinted tissue behaves in the real way as like a native tissue, because you can create a lot of artifacts, and this sort of shows you the parameters. Here you see a manifestation, but you put sodium nitroprusside, it's a manifester, it increases it, and then LNM, you again suppress it down. And we also do some morphological with ECM. I know that I don't have maybe not much time. I'm going to go over quickly some of the other things which you're also looking at, and then focus on some of the applications. You're also looking at myography a little bit to look at the stress, and so we are looking at relaxation and contraction using our bioprinted. So what I want to show that we are able to bioprint this tissues, and then able to characterize them and able to maintain them in a nice environment so that they can be viable for 15 to 20, 30 days. And so that's one of the main goal of these applications. And as I said, I focus mostly on the cardiovascular applications. We are also now starting this neural tissue models, as you could see here, using iPSC cells And so that's one of the project we are right now starting, looking at neural cells and looking at the blood-brain barrier and all those things. And what we're heading there in parallel, we're working on an organ chip model, which is more complex. But that's all we're ready to do, is to like a microphysical system where you can measure all these, parameters in a organ on a chip system. And just want to quickly touch base. What is the motivation of our cardiovascular research? We are looking at space, because when you go to space, because of the radiation and the microgravity, so we have actually already done enough experiments on our simulators. We mounted these samples on this microgravity simulator to look at the cardiac stress, because you're looking at the oxidative stress. That's the reason I introduced you that why are you looking at these biomarkers. So that's one of the main applications. And as I also mentioned, that what you like to do when you have an increase in oxidative stress, we are going to look at some angiotensin-2 receptor blockers such as losartan to see if you can reduce the effect of oxidative stress. So one of our research right now is focused on looking at the microgravity and radiation environment. We are going to start with simulated experiments right now to look at this oxidative stress, and hopefully then we can use this receptor blockers to prevent this oxidative stress. So that's sort of our goal. And thank you again for your time.